Hi, everyone. My name is Mark Stebbin, President of USCI Canada, and I'm very happy to welcome you to this webinar. Today, we have a very interesting subject, curing HIV. How close are we? And I have the privilege to introduce Dr. Santiago Perez Patrigeon from Queen's University and KSAC. The webinar objectives to understand the concept of functional cure versus sterilizing cure, learn how many people have effectively cured of HIV and how was this achieved, be familiar with the different cure strategies currently being used to cure HIV, and to be aware of the latest findings in HIV cure research. And for this webinar, we have for speaker an interesting researcher. Dr. Perez is not only an infectious disease physician with a clinical fellowship in HIV and AIDS, but also a PhD in HIV-Immunology from Institut Pasteur in Paris, France. Dr. Perez worked as a clinician-researcher at the Instituto Nacional de Ciencias, Médicas y Nutrición, Salvador Zubirán, in Mexico City for 10 years before accepting a position at Queen's University. The main interest is translational research on infectious immune-pathogenesis, mainly HIV, as well as clinical trials. Since the SARS-CoV-2 pandemic, they've participated in randomized controlled trials investigating new antiviral and monoclonal antibody therapies to treat COVID. So it's my pleasure to pass the microphone to Dr. Perez Patrigeon. Please.
Thank you so much for the kind introduction. And honestly, thank you so much for the invitation. When someone asked me, I think it was Brad, no, can you speak about HIV cure? Of course, anytime. I would speak about this for weeks in a row. We only have 1 hour, and that's a shame. And I'm also, even if I appreciate so much the invitation, it's too bad that we have to do it over Zoom. I really miss the face-to-face interaction, although I recognize that this Zoom technology and allow us to speak to a broader audience all around Canada, and eventually with the internet, I guess, farther away. So without further ado, let's talk about this. I wanted to go to the next slide. There you go. So just a bit of conflict of interest disclosure, that there's no conflict of interest that I have to disclose at this moment. And I did want to acknowledge a little bit the land, maybe say that I know that this is an unceded territory, of the Haudenosaunee and Anishinaabe peoples, and maybe acknowledging that just by simply stating this, it's not changing many things, but honestly, hopefully it works as a reminder that things need to be changed, and change is coming. So hopefully we can hope for a peaceful and better future. So okay, we're talking about cure. So as I said before, as it was mentioned before, we have two kinds of cure. There's this sterilizing cure, and then there's the functional cure. So what does it mean? So in simple concepts, we have this thing called the latent HIV reservoir, which is where the HIV is hidden, and we'll see that HIV is a retrovirus that integrates its genome into lymphocytes and some other cells, and that's what we call the reservoir. So we have this HIV latently infecting many cells, and the idea, very simple, is to do something about it. So whether it's gene editing or latency reversal, many things that we'll discuss about today, and then we end up with, as we say in Spanish, nada. So nothing, the reservoir is eliminated. We have absolutely no traces of HIV in the body, and that would mean absolute cure. Now, maybe we can't achieve that absolute cure, right? Maybe we can have something that would be like a cure, but without eliminating or without the need of absolutely eliminating HIV from the body. So we'll have the same situation, latent HIV reservoir. We do things, and then we end up with a smaller reservoir, and which allows the natural immunity, no, let's say NK cells or cytotoxic T lymphocytes, no, mainly CD8s, for example, and antibodies that readily control this reservoir and do not let it proliferate. So without the need of treatment, we would allow the patients and HIV to coexist and not having any signs of the disease anymore. So that would be, you know, kind of a good thing, even if it's not sterilizing cure. So first and foremost, I think one important thing to discuss would be, is this really necessary? So at the moment, we have extremely effective treatment. It was mentioned before, no, at the end of the 1990s, no, that it was called HART because it was highly active antiretroviral treatment. We've since then moved away from that nomenclature because everything is so effective that we only call it ART, A-R-T. HART wasn't that bad as an acronym, no, because it made you feel like that I'm giving you heart. Wow, it's such a warming analogy, no? I'm giving you a treatment which will save your life, and I'm giving you the heart. So anyway, but we did move away from that, and now it's not highly active. It's extremely active, extremely effective, so extremely effective that even we're going back to monotherapies, right? We went from not very effective monotherapies to somewhat better effective dual therapies, and now and we had to move on to the highly effective, which were triple therapies. Now it's so effective that even we've circled back to sometimes monotherapies. Even if it's triple therapy, we now have all those three combined in one single tablet regimen, or now even injectables, right? Every 1 or 2 months, and probably subcutaneous implants soon to come. And we know that by having this simplified regimens, we allow the viral load to remain undetectable. And we do know, and this has become very famous since that if we remain with a non-detectable viral load, we do not transmit in HIV. And this is a very strong concept. U equals U, undetectable equals untransmissible. This is very important to remember. And also, it's almost tolerated by everyone. Like everyone who takes this single tablet regimen tolerates the medication. We know, and I put this here almost. I mean, it's not entirely true. Some people do experience toxicity, of course, and there's questions about the metabolic impact of certain antiretrovirals. There's questions about still having this chronic immune activation, which ends up or participates in cardiovascular disease and cancer, for example. So there's still things going on, but honestly, we could say it's almost 100% tolerable. More and more, we are able to diagnose earlier. No, there's tests, ELISAs, PCR, etc., that are done and have become more and more sensitive. So we can diagnose as early as 5 days in the earliest. No, maybe. Although most frequently it's about 2 weeks, and for the very early diagnosis. But usually we do diagnose over a month or 3 months. Previously, it was 10 years before we could diagnose the HIV, right? So that helps. If we diagnose early, we can stop the replication earlier, and we can stop transmission earlier. So that's really good. And we also have better tools to provide effective prevention, no? Such as giving treatment before exposure, or even treatment right after the exposure, both PrEP and PEP. And well, we also have barrier methods, which have always been there, or I mean, for such a long time. And we know that if they're used, when they're used, they can be useful in certain situations, which is also part of the prevention methods. Anyway, so why do we use it? Well, from an individual point of view, speaking about cure, and I've spoken to patients about this. I mean, it is important, because even if we have so wonderful tools such as this extremely effective antiretrovirals, well, there's issue with access. I mean, it's good that we have these on hand, but if I can't access it, then how am I going to be able to take it, right? So it is a problem. Access is a problem. Even in Canada or in the States, there are access issues. People don't have access to all medications. It seems unthinkable, no? In a healthcare system such as Canada, well, yes, some provinces do not cover everything, or at least not to 100%. So patients can have access to certain medications, but not all of them. Anyway, every province has its own rules. Also, in certain countries, the government buys the whole the antiretrovirals for everyone. This is the example of Mexico, for example. But still, there are many barriers for the patients to go and get this the healthcare and be engaged into care. So there's issues with access. I also discussed already about the cardiovascular disease, kidney disease, metabolic disease, and cancer, which are related to this chronic immune activation or related to toxicity of the antiretrovirals. It is important to consider. We have an aging population of patients living with HIV, and as people age, of course, they're getting more and more comorbidities, and as such, they have to struggle with interactions, and they have to struggle with tolerance, and still, of course, as always, adherence. And if you don't take the medication well, then you end up with resistant virus. And that's another problem of current antiretroviral therapy. If you don't take it well, or to 100%, then you end up with a resistant virus, and we end up in square one again. And last but not least, sadly, I feel that it's the worst pandemic of them all is stigma, because stigma messes with everything. Stigma prevents people from getting the healthcare they need. It prevents people from even going and ask for help. Like, they don't even want to get tested because they're afraid of what other people might think if they're getting tested. They're afraid of taking medications to prevent HIV because of what other people might say or think. And in certain parts of the world, it may be only discrimination with words. No, maybe we won't talk to that person because they may have or may not have HIV. But in certain other parts of the world, it's a life or death threat. So it's really awful. Like, people are afraid of even disclosing that they are interested in in having a diagnosis or ruling out HIV, and because they might end up dead, right? So it's really awful. Stigma and discrimination, I think it's the worst of the pandemics. From a public health point of view, there's still a lot of work to be done. This is one of the latest fact sheets of the UNAIDS. And as you can see, there's, I mean, the targets for 2025 have been revisited, no? And we're aiming for the 95, 95, 95, which means, as you know, we need to diagnose 95 of the people that live with HIV. We need, out of those people diagnosed, we need 95% of them engaged in care and on treatment, and we need 95% of those with an undetectable viral load. But we're not there yet. Many people are not, even if the numbers are increasing, which is a good thing, this number here that shows only 86% know their diagnosis. That means that 14% do not know their diagnosis, and that means they're not engaged to care, and they're still, of course, transmitting the disease. And so it is a barrier for us to eradicate this infection. So many obstacles we face to try to cure HIV. On the right side of the slide, you can see the life cycle of the virus. As you can see, of course, this is an RNA virus, and the most important interaction is GP120 that interacts with CD4 with the CD4 protein, and CD4, which acts as a co-receptor for the T cell receptor, folds and allows GP120 again to interact with another receptor. It's called co-receptor, so it's a CCR5 or a CXCR4. There are many others, but those are the main ones that HIV uses. Once GP120 interacts with this second receptor, it has a conformational change. It opens and allows another glycoprotein, GP41, to unfold and inject itself into the cell membrane. And then this GP41 acts as a, how do you call it, a tie, a bow tie, I think it, and it twists and it allows the two membranes to get together and fuse. Once the fusion happens, then we have this reverse transcription, which changes, of course, from an RNA copy to a DNA copy. And this DNA copy gets into the nucleus and is integrated thanks to the integrase, of course. And then the process, once it's integrated, then as I told you, it becomes the part of the reservoir. And after that, in order to replicate, the virus needs to transcribe its genes and introduce, of course, the proteins, which assembly, the protease is used at that moment, and then they bud, no? An immature virion buds out of the cell and then matures outside. So we can inhibit each step of the cycle with treatment, and it's important to know. So about the obstacles, I discussed the human factor. So the fact that people don't even want to engage in care because of afraid of stigma. So that's an important factor to consider as a barrier for cure. We have this persistent viral replication. So that is something that we have to deal with. Even people that have a non-detectable viral load, there may, in certain situations, they may have persistent viral replications to a very low level, and that repletes the reservoir. And again, speaking of the reservoir, that's the true enemy here. It is a true target, no? If we want to cure HIV, it is the reservoir. And knowing how to measure the reservoir, knowing how to attack the reservoir, that's something that will allow us to achieve that HIV cure. there's also a problem. There's what we call an anatomic reservoir. So HIV not only can, of course, hide in the nucleus of certain cells, but also those cells may be located in certain parts of the anatomy that are not that accessible to certain medications. For example, the brain, no, that could be one. Oh, I repeated the slide. That's good. There you go. So let's talk about the reservoir and how the reservoir is made, no, and how it's composed. And so if we review the acute or the processes that happen during the acute HIV infection, we would know that the reservoir is established immediately after the infection, like very early after the infection. There's a period of the early infection that is called the eclipse period, that it's not a true active infection yet. HIV is in the virus, in the body, sorry, but it's not, it has not yet caused or produced an active infection. The first cell in this model of mucosal infection, the first cell that HIV encounters is a dendritic cell. And dendritic cells are trained, of course, to capture antigen. Once they capture antigen, they're activated and they mature and they migrate to the lymph node in order to present their antigen to the lymphocyte. The problem with these dendritic cells capturing HIV is that they englobe it in these vacuoles, but HIV does not get processed. It lingers there, hiding in those vacuoles. And I'm sorry, some of my slides I noticed that they have Spanish words. I've given this lecture in Spanish sometimes. So this process where HIV encounters a dendritic cell, and then this dendritic cell captures antigen, captures HIV, activates, matures, and migrates, takes a while. As you can see here, even in Spanish, I do apologize, it takes hours to days. And that's why during those first 3 days, we call this period abortive, because if we provide treatment at that moment, we can maybe stop this and abort the infection. And that is why we use PEP in those first 3 days, because the infection is not really there. Then we call that, I mean, in the next phase, we call this an occult infection, because even if it's too late, it's irreversible, it migrates to the nodule, and the virus is infecting these new naive T lymphocytes, but there's not virus yet in the bloodstream, no, causing a detectable viral load. So that's why it's occult. We are not able to diagnose it with the current methods. And then once it's an established infection, then the viral load increases, of course, and there's detectable P24 antigen, detectable PCR, and of course, it extends to everywhere else in the organism. And as I said, the reservoir can be hidden or can be composed of several T lymphocytes, particularly central memory cells. But these cells may be distributed in different anatomical sites. And that's why the cellular distribution of the reservoir is important to take into account when we are trying to cure. No? So we can have it in lymph nodes, we can have it in the central nervous system, we can have it in different tissues, such as lung, skin. There's also the adipose tissue that has been described as yet another reservoir. And the main, of course, tissue where everything happens is the gut-associated lymphoid tissue. But of course, the proportion of the different tissues into this big reservoir pool is different, right? So the gut-associated lymphoid tissue would be a major component. The lymph nodes would be a major component. The peripheral blood is also one important component, and maybe the adipose tissue would not be as relevant, but still relevant, because if it only takes one infected cell to re-establish that infection once the treatment is stopped. So that is one of the major challenges we face. So in order to review the different strategies that we have to achieve a cure, we'll go one by one. So first one, there's the stem cell transplant that has been tried and has its pros and cons, and we'll review them bit by bit. We'll talk about this new gene editing method, which is called CRISPR-Cas. And there's this shock and kill, and then shock and block, which is different, or lock and block. So first, stem cell transplant. So as we know, 5 people have been cured. It's 5 or 6, I get lost. So we know some of them by name. I mean, Timothy Roy Brown was the first one. And this slide I stole from the International AIDS Society presentation in 2023, which was last summer. And a good friend of mine that works at Institut Pasteur, Ciencias in Lyon, presented this in the cure workshop. And so Tim Roy Brown was the first one, and then followed by what we call the London patient. There was the Dusseldorf, the New York, and then the City of Hope. And there was a 6th one which we'll talk about later. So anyway, all these have in common having received stem cell transplant because of different diagnoses, so mainly AML. And they received this transplant with a donor that had a certain characteristic. They had this deletion in the position 32 of the CCR5 gene. So as I told you, CCR5 is one of the co-receptors of HIV. So it turns out that certain strains of HIV prefer CCR5 and certain strains prefer CXCR4. We call that tropism because in vitro, those 2 strains behave a bit differently. One has more tropism towards lymphocyte, and one has more tropism towards macrophages. But in vivo, it doesn't matter. It's just, I mean, they still behave somewhat the same. What we do know from a clinical point of view is that in the acute setting, we find more R5 strains, and in the chronic late stage setting, we find more X4 strains. Why is there a change in the tropism? I don't know. And well, there's many hypotheses, but we're not sure yet. It may be because CCR5 expression changes over time because of HIV. It may be for different reasons. What we know is that you most probably are infected with both, and then one takes over the other. Sorry, there you go. So yes, these are the times post-ART interruption. So all of these patients received their stem cell transplant and then ended up interrupting their antiretroviral regimen, and with different times, of course. It was decided, I mean, they looked everywhere and was decided that they were likely cured. Okay, this is not working. There you go. And I want to take a few seconds of your time just to reflect on Timothy Roy Brown, who passed away sadly, because of his leukemia. He had become very well known as the first patient to be cured of HIV, and he became very active and giving lectures all over the world. And he became an inspiration for the cure of HIV, and he became a symbol. And it was very sad to learn of his passing. Okay. So then happened the six patients, and this is very interesting. This was a news of last summer, and this patient received a stem cell transplant, but without, I mean, the donor did not have the CCR5 mutation. And this is very important because it means that with the stem cell transplant process, of course, the chemotherapy, the radiotherapy, etc., we are killing the reservoir. But patients, there are many patients who undergo that treatment and they're not cured of HIV. And that's why it was thought that maybe you needed that extra delta 32 CCR5 mutation, no, for you to really become cured. And turns out that this, at least it's a proof of concept, that it may not be true, that you could end up cured if you undergo a stem cell transplant without the mutation. And again, just to remind you that the CCR5 is the core receptor of HIV. It interacts with GP120, and it is the one responsible for the GP120 change in conformation and exposure of the GP41 that allows then the two membranes to fuse. So this is part of the abstract that was presented and again presented by us here. And so after 18 months, there was no viral rebound, and we'll see some of the findings, no, that he presented. And this is all a very interesting effort for this. This is a consortium. It's the E-System study group. And mainly it's people from Europe, but it's such an important collaborative group. What they do is they're not promoting stem cell transplant in HIV, not at all. It's just that they say, well, okay, we acknowledge that certain patients with HIV need to undergo a stem cell transplant. So if you have a patient that has these two conditions, HIV and the need for a stem cell transplant, please give us cells. No, so it's a kind of a protocol, no, where you know exactly how and when are you going to retrieve certain cells. So they do apheresis and many other cells, and the E-System group takes them and analyzes them with PCR, of course, ultra-sensitive tests. So this is the story of the more in detail of this patient. The Genova patient. And so 20 months after the stem cell transplant, he had this patient had no detectable viral load. This is a male that was diagnosed in the 1990s. He had become suppressed on antiretroviral therapy since 2005, but then had sarcoma in 2018 and had to undergo chemotherapy and total body radiation, and finally ended up with this stem cell transplant in 2018. He had chimerism, and then something very important, and probably it's the key, is that he had acute and chronic graft versus host disease. And this is something that hematologists know about for a long time, is that when a patient experiences graft versus host disease, sometimes this helps cure their leukemia, because the patients experience graft versus leukemia, right? And in this case, maybe what they experience is graft versus HIV reservoir, right? So the CD8s from the donor take over and kill all the remaining cells which were infected. No, of course. Again, we're not suggesting the graft versus host disease is a good thing. It may be very toxic, and people die from graft versus host disease, but it may help explain what happened to this case. No, so this is just graphs showing the viral load that was undetectable even months after the stem cell transplant, how the CD4 CD8 ratio evolved, and how the CD4 T cell count evolved afterwards too. And these are all the tests that were done looking for HIV in this person. And as you can see, with even ultra-sensitive viral loads, measuring DNA, HIV DNA in peripheral blood cells, there was no HIV found. Trying to look at it in CD4 T cells, and even in a bone marrow sample, there was no HIV found months after the transplant. So it's impressive how we lost completely evidence. And look, they looked everywhere. They looked in the ileum, the cecum, the ascending, transverse, descending colon, sigmoid, and rectum. They looked everywhere. They biopsied everything. And with that, no amplification detected. So they really made sure that that's why it takes a long time, and it takes many people and many research groups to really identify and confirm that a patient has been cured of HIV. So in summary, stem cell transplant, 6 people have been officially cured of HIV with and without donors that had or that carried the Delta 32 mutation in the CCR5 gene. So that's the truth, that's what happened, but nobody is proposing to perform a stem cell transplant in every HIV, in every patient living with HIV. That would be a mistake. It is a very risky procedure, sorry for the typo. It carries mortality still, no? And many people that do need it and undergo this treatment die. It is unpractical and costly. So it's not applicable for the general population. And applicability and generability, that's something we need also, no, when we propose a cure. Now, acknowledging all these defects, we do need to know that it is the only thing that has worked so far. Okay, let's move on with the next. So shock and kill. So let's talk about latency. I told you already that HIV infects cells, and then its genome gets inserted into the human genome, and that's what we call a latent infection. So if the cell is dormant, the HIV genome will stay there dormant forever, for as long as the cell lives. So that's the problem with central memory T cells, because they live forever as long as the human that carries them lives. And how they manage that? Well, they have their own way of having a homeostatic replication. So they do replicate, but very slowly. They have this IL-2 secretion, and so they remain alive, and thus repopulating the HIV reservoir that they're carrying. Once these latent cells activate somehow, for some reason, there's many reasons that a cell can be activated. It could be a general activation of the immune system, or it could be that one of the genes, for example, TAT, just simply decides to wake up and replicates. So anyway, when the cell becomes active, then of course we have an active replication. And here's where the cell is at most vulnerable, because it starts to express viral proteins in its membrane. And that's where it's recognizable from CD8 T cells and NK cells, which ultimately destroy that cell, right? So actually that's the opportunity, because if we somehow activate latent cells, right, or stimulate transcription somehow, then we may be able to recognize these latently infected cells. Now, of course, that also fuels HIV replication, so that's dangerous, and that's why we need antiretroviral drugs. This is a little bit like giving money and training to a certain militia group in order to, in the future, overcome certain governments. I'm not saying this has been done anywhere or at any time in the history of human race, but it's just an idea. So of course, this has been done. The problem is that it doesn't work very well. So far, we have many drugs that have proved effective in shocking cells and in stimulating replication, but when we measure the reservoir, there's no difference. So I'm not going to discuss how frustrated I've been with this strategy. There's another new strategy, which is maybe an equivalent, but a bit of a twist to this shock and kill, which is block and lock. So we do activate replication, but at the same time, we block transcription. So this is important. We have inhibitors of this transcription initiation, and that is a good idea actually, because it allows HIV genome to stay integrated, but forever. So in a way, we trapped HIV within the cells. The idea is that if we don't need antiretroviral therapy, if it remains in our genome, it doesn't matter to us. We won't be cured in a sense that we don't have any HIV in our body, but we would be cured functionally because HIV in our body would not be able to replicate. So that's block and lock. It's a good idea. However, it's still on the works. It's still very preliminary, and we don't have evidence yet that it has produced significant impact in the reservoir. But I think it's a good idea. The one that's more promising, and I'd like to maybe discuss more about it, it's CRISPR-Cas. So that's gene editing, and this idea, was suggested and was born from the works from Francisco Mujica in the 1990s, and then Feng Zhang more recently. And the idea is that, we have this RNA, with transporter RNA that is linked to this caspase, to this enzyme, right? So the RNA recognizes a portion of DNA, and the enzyme cuts the DNA exactly in that portion. We can even twist it and help the repairing process. So we can induce mutations in the genome, specific mutations. So if we cut HIV genome, for example, we cut NEF, then the virus will not be able to replicate. Or if we cut TATs or GAG, for example, then HIV will remain, but it will not be able to replicate. So that's the idea with CRISPR-Cas. And this has been actually has proven to work in vitro. We have already evidence that cell cultures with HIV that have been treated with CRISPR-Cas have become completely cured of HIV. I mean, we don't call it a cure in a in vitro model, but you get the idea. We have eliminated HIV from that culture, and this is a good thing. And we can also, of course, edit the core receptor at the same time, blocking viral entry. So there's many strategies using this CRISPR-Cas. It has now also been used not only in vitro, but in vivo in a humanized model. So in this publication, this was actually communicated in 2019, and it's called laser art. We see that mice that have been treated with this CRISPR-Cas end up having no rebound in their viral load. Now, you can tell me, okay, this is not absolute, this is 50%, and I understand. So it's not 100% foolproof, but at least it gives us hope that it may be something that can function, no? And this is with laser art and then sequencing to dual CRISPR. So it's combining two treatments, laser and CRISPR, and we see that we have also evidence of cure in this mice. So we are not only targeting the virus, but also targeting the CCR5 gene. And this is the one-shot new approach, and again, acknowledging the work of these two people. Other therapies, well, there's been lots of work ongoing. There's investigation about homeostatic cytokines such as IL-2, IL-7, trying to boost CD4 recovery, and at the same time boosting HIV replication. We know IL-7 does that, but it doesn't seem to work very well, and honestly that has been maybe stopped. We also, thanks to the cancer research, we have seen that inhibition of certain markers that we call checkpoints helps a lot in reactivating cytotoxicity. So for example, a cell that expresses PD-1 starts to not be able to secrete interferon and not perform well. And we call that exhaustion. And so if we inhibit PD-1, there's evidence that this cell will start to recover this capacity, of secretion, interferon secretion. And so if we do that, well, cytotoxic cells might again recover anti-HIV activity and thus help controlling the virus. And there's some evidence, well, and they use that for anti-cancer immunotoxicity, so that it works well for certain cancers. So for HIV, there's certain papers showing some promise, but at the same time, some not. It's discrepant. So more work needs to be done. We were a few years ago, we were very excited about the anti-alpha-4 beta-7, which is a marker, a homing factor for the gut. And so when it was used, this alpha-4 beta-7 therapy, some monkeys were cured, but then it was difficult to replicate, and so we didn't think that it was a good strategy. And now, more recently, there's the CAR T cells, which is again a strategy used to cure leukemia. So we take out CD8s, we train them to attack certain proteins. So, for example, HIV proteins such as GP120, and then we infuse them again, and that will help to treat or to attack the reservoir. Problem is reservoir cells, as I showed you, do not express GP120, for example. So as long as they're remaining latent, then they won't express this and they will be immune to the CAR T cell therapy. So that is still on the works and also is extremely expensive, but we're hopeful for the future. And then there's the BNAbs. BNAbs stands for broadly neutralizing antibodies. So that's another strategy that is being used. If we treat with BNAbs, so these are, again, broadly neutralizing antibodies. So if we treat with BNAbs monkeys, for example, and we infect them with HIV, they are protected against the infection. But all these models, they give BNAbs very early or even before the infection. In a real life scenario, we don't use it that way. No, I mean, it's really hard to go to the hospital and receive a very expensive intravenous therapy and use it as the pill for the day after. No, we can't propose that. So it's really hard. The ideal would be to infuse these BNAbs and they would remain for a long time in the body. So it's meant to be a preventative measure. But of course, HIV mutates so much that it becomes resistant to these BNAbs, and we're back to square one again. So in summary, understanding the reservoir, knowing your enemy, that's the best strategy that we can have. Combination of strategies is key. So using preventative measures and then combining with good antiretrovirals and then also using strategies like, for example, gene editing and editing the CCR5 plus editing the GAG. That's a strategy that may be helpful. And we have to think, when we think about HIV cure strategies, about the scalability, like about the generalization of this proposed method, no? Because we don't want to cure 6 people, we want to cure everyone. We need to cure 40 million people, right? So that definitely will be a challenge. But if we continue to cure one at a time, it gives us so much learning, no? We learn each time so much so that we can maybe at some point achieve this. So how close are we? I think we're much closer than 30 years ago, but there's still a long way to go. And with that, I really thank you so much. If you have more questions than the ones we'll discuss right now, you can follow me on Twitter. It's not even called Twitter anymore. I know it's X. But and if not, you're more than welcome to visit us at Kingston, Ontario. And if I may, we're organizing the first HIV symposium and STIs in Kingston, Ontario, and Brad Stoner is one of the members of the organizing committee. So you're more than welcome. It will be June the 20th.
Good. So thank you very much, Professor Perez Patrigeon. I think we scratch a lot of ideas on the dartboard, and maybe I have a short question. How would you compare the cost of doing these technologies compared to the cost of using art, since art is so good?
Yeah, no, that's a very good question. And I actually goes back to the first questioning, my first slides, is this really necessary? So using art has become less and less expensive. No, it was really expensive at the beginning. And for example, when using art as PrEP, we always joke that it's a very expensive condom, right? Condoms are so cheap. And so if we give PrEP to people, it works even better, but it's much more expensive. And trying to give antiretroviral to people when they don't even have it for treatment, it's really tough. So now we're proposing other cure strategies which are 10 times more expensive than antiretroviral. So is it worth it? I think we have to push the boundaries, and we have to keep pushing. And pushing is expensive, that's true. But once we know better and we pierce, and move forward, then we'll be able probably to cut costs and make it affordable for everyone.
Thank you. Someone has a comment. Are you saying that we literally find a cure for HIV? It appears that sterilizing cure or viral load suppression is not entirely cure, but treatment.
Yes, exactly. So the concept is viral suppression requires continuous suppression, right? So it requires antiretroviral therapy for as long as you live. What we want is the same, but without the need of treatment. We want that just one-time thing, and then you are cured. So sterilizing and functional cure both would achieve the same. The only difference is that sterilizing cure, you would not have HIV in your body. Functional cure, you'll still have HIV in the body, but it will not do a thing. HIV would become like the herpes simplex of the 21st century.
Thank you. There's a comment. We didn't hear so much about therapeutic vaccine. How about therapeutic vaccine? They were supposed to be going somewhere.
Yes, so thank you. I mean, as I said, I only have 1 hour. So there's lots. The idea of the vaccines is to induce naturally broadly neutralizing antibodies. So if we can teach your body to produce broadly neutralizing antibodies, then HIV replication would completely decrease, and you would prevent infection, and also you would end up with HIV cure. That's the idea. What we know about that is that if we use broadly neutralizing antibodies to chimpanzees that have been infected for a long time, we don't achieve cure. If we use them very early after the infection, then it seems to help. So in a way, it's more preventative than curative, than therapeutic. So maybe that's why you haven't heard so much.
Thank you. And maybe a question from the herpes field. You were talking about herpes. Why are we talking about sterilizing HIV? We're not talking about sterilizing HSV.
True. Well, herpes simplex is uncurable, as you know, but it's not that bothersome for many people. Like you just forget about. Many people are infected and they forget about it their whole life, right? So we achieve that functional cure. We don't need treatment for as long as we live. A few people do require suppressive therapy, and so that with that they do not transmit and they don't get as many recurrences, right? So yes, we are trying to cure HSV, and we have new antivirals, but sadly, none of them have achieved a sterilizing cure. So we have settled with functional cure for HSV, which is you give cyclovir or valacyclovir, and it works with 1 week of treatment. No, it usually works. With HIV, you can't do that, right? It's so much more harder and so much more difficult.
Thank you. Why not strive to make suppressive therapy delivered by infrequent treatments?
Yes, we are working on that. So now, as I told you, we have injectables, which is 1 month or even 2 months, and soon we'll have subcutaneous implants. So it will last 6 months or even a year, or why not 5 years? No, so that is in the pipeline. So that's one strategy. Now, remember, discussing with people living with HIV, this does not get rid of the stigma. So once you're cured, you don't have HIV anymore, no? And that frees you from that stigma. And that's important, very relevant in countries in Africa where stigma can get you dead, no? Can get you killed. So it's really relevant to be free of the disease. So that's why we still are hoping for a complete cure. But of course, pharmaceutical companies are looking into prolonged therapy, and I think that's the way of the near future.
Thank you. Someone expressed a comment about HSV is very bothersome for many and newborns, and that there may be a misstatement about how many need HSV suppressive therapy. So I see maybe in the near future that we have a debate, HPV in one corner, HSV in another corner, hepatitis C and HIV.
Yes, I'm sorry. This is just like Twitter, right? We can't discuss with so much detail in so small characters, no, so few characters. So yes, I apologize if I maybe undermined how many people need suppressive therapy. I understand. I treat many patients with suppressive therapy. So yes, of course, probably in the proportion of people infected with HIV, the proportion that requires suppressive therapy is less than the proportion that patients with HIV require suppressive therapy, right? In the case of HIV, it's 99% of the population that lives with HIV that requires suppressive therapy. In the case of HSV, it's much less.
Thank you. So we have a last comment. There's a group, a new organization called Herpes Cure Advocacy, and this is led by Jeff Klausner in California. So maybe we'll invite Jeff to talk about his thought about herpes cure in a near future. So thank you very much, Professor Perez Patrigeon. Very nice, very interesting, challenging discussion we had. And I hope we have time to sit and discuss longer at another meeting. So just to let you know, there's a membership drive for IUSTI Canada. You can go on IUSTI Canada website and register as a member. Being a member will also decrease your costs for registering at various meetings. So one of the next international meeting will be the IUSTI World Meeting. That's going to happen in September in Sydney, Australia. Registration is opening. And I remind you that IUSTI Canada is behind the making of the 2025 World Congress of ISSTDR IUSTI, and this will happen in Montreal in July 2025. So being a member makes a good discount on the registration price. So thank you very much, Dr. Perez Patrigeon. Thank you, Brad, for bringing us to our attention the work of Dr. Perez. And thank you, Greg, for the help behind the logistic. So you'll have soon information about the meeting that will occur in March. So for IUSTI Canada, I thank you for being present. And for those that want to rewatch or want to send a link to their colleague that could not be present, the link will be on IUSTI Canada as the link from other conference we had in the past. So thank you very much and hope to see you soon. Bye-bye.